thank you, everybody, and thank you especially to Professor Boris Chaumette, which is you probably have had time to read all his appointments and what he has been doing in Canada and in Paris. I wanted to share what is not there. Boris works in the coolest place that you can work in, which is in Hospital Sainte-Anne in Paris. It's a wonderful, very historical building where the amount of history there is extraordinary. It's where Talleyrand started to describe the atlas of the brain. It's where Delay and Denneker started the very first psychopharmacology for schizophrenia, where chlorpromazine was ever done. And they have a fantastic, not only these historical clinics, but also a fantastic new buildings for research. Boris not even is working in the coolest place, it has the coolest of all the coolest of the clinics that you can imagine. He has set up a national center for rare genetic psychiatric diseases. Is a place to go for understanding some of the genetic markers that are associated with the onset of psychosis. So for me, it's an absolutely privilege. It's an honor to have here today with us Boris. And I think that we will have, as usual, the 45 minutes of talk, and then the Q&A. If you want to send all the questions in the chat, we can catch up later. The floor is yours. Thank you very much. And Boris, welcome.
Thanks a lot for the presentation and the kind invitation. Unfortunately, I cannot make it to come today to visit you, but it's a pleasure and honor for me to present part of my work. So as Emilio said, I did most of my research in Paris, but I used to work in McGill University, also with colleagues working on neurogenetics. So I have both expertise, both sides of the Atlantic Ocean. That's always cool to have also feedback from colleagues all over the world. So I hope that it will be interesting for you, but I hope that you will also suggest me new ideas and to comment also the results I will show you. So since my PhD, I'm working on the biological factors associated with the onset of psychosis. But so it's in Paris, we are based in the GHU Paris, which is the new name of the Centre Hospitalier Sainte-Anne, Hôpital Sainte-Anne, which is one of the historical hospital directly in downtown Paris. And as Emilio introduced that to you, we also built a new building called the Institute of Psychiatry and Neuroscience of Paris, which is just in one building in the hospital. So it's a very cool place to do clinic and research in the same time. So patient can circulate from the hospital to the research part, and we are collecting samples for doing research in molecular psychiatry. So as you may know, schizophrenia is a neuroprogressive disorder, not starting immediately after birth, but there is a vulnerability during childhood and adolescence, and then during late adolescence, people can have at-risk mental states with attenuated symptoms, and then around the 15 to 25, they can have first episode of psychosis, and then the illness become chronic with relapse and sometimes also resistance to treatment. So that's a new paradigm developed since 2-3 decades now, and according to many colleagues in the world, that's the natural history of schizophrenia. Another main idea in schizophrenia is that the duration of untreated psychosis is associated with clinical and functional outcome. This is based on the meta-analysis by Marshall and al. In 2005, showing that the later is the duration of untreated psychosis, the worse is the outcome after 1 year. In terms of all kinds of symptoms, depression, anxiety, negative symptoms, functioning, positive symptoms, quality of life, all these symptoms become worse when you start treating the patient after a significant delay. So it means that early intervention could be the key to drastically change the outcome of patients in psychosis. The question is, when should we intervene? Should we intervene in the first episode of psychosis, or should we intervene when the disease is already chronic, or should we intervene at very early stage during at-risk mental state. So that's a debate because at-risk mental state is a category, diagnostic category with uncertain clinical significance, because if you take patients that are help seeking in the hospital, for instance, or in general practitioner and psychiatrist, some of them will have other psychiatric diagnosis. Some of them will be totally healthy, just asking for some exam from the psychiatrist. And we estimate that around 1/3 of them will have a high-risk mental state. So that's pretty much. And these high-risk patients did not always evolve to full-blown psychosis. Some of them will develop other psychiatric disorders like substance abuse, depression, anxiety. Some of them will have persistent attenuated psychotic symptoms for over their life, like it was called before the schizotypic presentation. And some of them will totally recover from this presentation. So we estimate that only 1/3 of these at-risk patients will evolve to psychotic disorders. And the percentage of patients evolving to psychotic disorders, which these patients are called converters to psychosis, we estimate that the percentage is maybe overestimated when we say that it's 1/3, because in recent studies, around 20 to 25% of them evolve to real psychotic disorders when we do the follow-up. So it means that we have an uncertain course of the illness, and it's not mandatory to go to each stage of the illness. And the question is why some patients convert to psychosis and why some patients are resilient. They mean that they don't convert to psychosis. And can we understand that? Can we intervene early in patients that deserve this kind of intervention? Or can we just be quite reassuring for the patient who will not evolve to psychosis? Another story is the question of the treatment. We know from a long time that olanzapine, risperidone, all these kind of antipsychotic treatments are not efficient to prevent the onset of psychosis. In this meta-analysis in the Cochrane literature, olanzapine did not succeed to prevent the onset of psychosis. So it means that if you give this kind of treatment, antipsychotic treatment, to at-risk patients, you will not prevent the onset of psychosis. But in the same time, which is very clear, is that you induce a lot of adverse effects. For instance, with olanzapine, you have a weight change that is very dramatic. So you increase the obesity, you increase the cardiovascular consequence with this treatment, and sometimes you didn't prevent psychosis. So in terms of benefit-risk balance, there is a very big issue because you have to treat a lot of patients with uncertain outcome, because not all of them will evolve to psychosis, with inefficient and side effect providing treatment that is worse sometimes than just leaving the history, natural history of the disease. So the question and the overarching aims we want to answer is, can we understand better the pathophysiology of psychotic emergence? This could help to develop new treatment, to identify new therapeutic targets, and to develop treatments or intervention adapted to the stage of the illness. We will not treat early phases of psychosis as we are doing for chronic presentation. At the same time, can we predict the outcome of at-risk individuals using biomarkers? Because if we have biomarkers, we can just at the very beginning just classify future converters, future non-converters, and intervene early only in converters. So there is many research on these topics now, and on my side, I choose to work on molecular biology because I really think it's a future. We can discuss that, of course. Imaging is cool, clinical and phenotypic assessments are very useful, but also molecular biology can really lead the way for the future of psychiatry, in my opinion. So I will present what I'm doing in this kind of approach. The first things we cannot just ignore is that schizophrenia is highly genetics. And that's known from ancient and old epidemiologic studies. For instance, if you have one monozygotic twin that is affected by schizophrenia, the risk for the second monozygotic twins is 50%. So it means that he has one risk on two to develop the disorders. So, of course, genetic matters, and this is also known from a patient related to an affected parent or siblings. And genetics is really important in schizophrenia. Schizophrenia and psychiatric disorders are the most genetic disease in humans. But the problem is that genetic of this kind of disorders are not Mendelian most of the time. If they are Mendelian, we are talking about other kind of disorders, which is very interesting because you can have a specific intervention on them, and that's part of my activity to receive patients with, for instance, chromosomal deletion or genetic mutation. And that's a very nice story to tell for the patients and the families and the colleagues. But that's a very, I mean, it's not the majority of the cases. On the other side, we have common variants, which are implicated in common disease, and we found them using genome-wide association studies. So the common variants in brown here are common in the population, because maybe 20, 30, 40, 50% of the population can share these variants. And the risk associated with the disease is very low. The odds ratio, for instance, can be 1.05, 1.1, 1.2 at the maximum. So this common variant, you cannot say based on that only that the patient will develop the disorders, but that could be an interesting point to do research. Because now we know from national and international efforts through the Psychiatric Genomics Consortium and a new study published this year, we know that hundreds of polymorphism of variants that are associated with the risk to schizophrenia. So for each chromosome here, you can see the p-value of the association. It's like we call that a Manhattan plot because it's like in Manhattan in New York. You can see the tower. Here, the tower are the significance of the association between one variant in one chromosome and schizophrenia. For all chromosomes, you can see that there is a dozen of polymorphism associated with the disease. And what we can do now is we can aggregate them into a score. And the score is, I put you the formula here, but nothing complicated really. You just have to look if the patient is carrying the polymorphism of interest and whiten this polymorphism by the odds ratio. So you can derive a score for each individual, and the score is representing the risk for specific disorders, in that case, schizophrenia. There is a very recent and exciting results from Jenna Perkins and colleagues. They use NAPLS, which is the North America Prodromal Longitudinal Studies, working on patients with at-risk mental states. And they compute the polygenic risk scores for these individuals. So they look at 80 converters, the patient at risk that convert to psychosis, 250 approximately non-converters, so at-risk patients that will not develop psychosis during the follow-up, and unaffected subjects. And they did that for European and non-European population. And what they found is that the converters have a higher polygenic risk scores than non-converters. And then ultra high-risk patients, they have a higher score compared to unaffected subjects. So it means that the higher your risk is for schizophrenia, the higher is your chance to develop schizophrenia, which is quite reassuring in the same time. And they also did a Kaplan-Meier curves of psychotic conversion depending on the polygenic risk score. So they compute the polygenic risk scores for each individual, and for the higher polygenic risk score, which is in green here on the right, you can see that the at-risk patient enter faster to schizophrenia than a patient with a lower score in terms of genetic risk. So it means that polygenic risk score is associated with the risk to develop psychosis in ultra-high risk patients. So we are involved now in this story to try to replicate and to extend the results and to do a meta-analysis of this, because we quite criticize the story, saying that it's very interesting in terms of understanding the risk, but it's useless in clinical practice. If you apply that to a new population, and we use the population of patient converters, non-converters in Paris, we saw that the area under the curve from the ROC curve is not significant. It means that we cannot use polygenic risk scores to predict the outcome of ultra-high-risk patients. And this is because when you are looking at the graph here showing in the abstract, you can see that the polygenic risk scores in converters is similar to the scores in non-converters. There is a big overlap, meaning that for a patient, for instance, at 0.5 of the polygenic risk scores, he has as much chance to develop psychosis as to non-develop psychosis. So we have done other study, and we published that last year in Professional Psychiatry. We did the same using the polygenic risk scores, and we applied that to other disease, and we tried to see if there is an impact on the intelligence quotient of ultra-high-risk patients. First, we saw that the polygenic risk scores for cognition is significantly associated and positively associated with IQ in converters and non-converters, which is quite reassuring, because the higher your polygenic score is for cognition, the higher is your cognition. Makes sense. And you can see at the same time that ultra-high-risk converters in red here have a lower IQ scores than non-converters for the same level of polygenic score of cognition. The second point is that we applied polygenic risk scores for schizophrenia to the same cohort, and we found that the polygenic risk score for schizophrenia has an effect, a negative effect on IQ only in non-converters, because the converters are more affected in terms of IQ, but it is not totally related to polygenic risk score for schizophrenia. It's similar to what we can find in general population. Polygenic risk scores for schizophrenia affect cognition in the general population, but did not affect cognition in a schizophrenic patient. Because maybe in schizophrenia, the patient has already some cognitive defect, which is independent of their risk for schizophrenia. So we are replicating the previous results in general population and schizophrenia cohort. And the last, we also look at the polygenic risk scores for resilience to schizophrenia, because you can compute now a score on for the resilience to schizophrenia, so the protective factors, protective genetic factors. And we found a positive correlation between this score for resilience and the IQ, especially in converters. This means that resilience did not prevent the converters to develop the disorders, but they increase their cognitive abilities when they are affected. So genetic matters. Genetic can be interesting in terms of care, because you can, using genetics, try to not exactly predict, but have a clue on which are the patients that will be more affected by schizophrenia or cognitive deficit. And maybe you can target this specific population for cognitive remediation, for instance. You can say, based on their genetic background, this kind of individual, for instance, those with a higher polygenic score for resilience will benefit more from cognitive remediation than the others. But genetic is not the alpha and the omega of the story. Because if you look back to the monozygotic twins, they share 100% of their DNA, but the risk for schizophrenia is 50%. It means that there is something more than only genetics. And we know that from several epidemiological studies, that genetics is at the beginning, but schizophrenia is developing during adolescence. So there is also environmental factors during the course of the disease, like infection during childhood, trauma, urban migration, social isolation, drug abuse, stress, and so who predate the onset of psychosis and precipitate some at-risk patients to a full-blown psychosis. If you look at the environmental factors and the odds ratio, the biggest odds ratio is for cannabis use, but also childhood trauma is significant, urban living, migration, and other factors like perinatal factors, obstetric complication, winter births, and so on. So we know the environmental factors, and we know that there is an interaction between gene and environment. This is based on a very old study related to the risk of psychotic disorders and urbanicity, which you can see on the right, on the green part, when the patient doesn't have siblings affected with psychosis, so there is free from any genetic predisposition, we can say, the risk related to urbanicity is at 2%. But when the patient is siblings of somebody who has psychotic disorders, there is a dramatic increase of the risk related to urbanicity. So urbanicity interacts with the risk of having a genetic, psychotic disorders, and there is interaction between gene and environment. We think that gene-environment interaction can be related to epigenetic mechanism. Epigenetic mechanisms refer to any change in the expression of genes not related to mutation on the DNA. There is several mechanisms like histone modification, microRNA, but also methylation of DNA. And this is methylation of DNA is one of the easiest way to explore epigenetics, because you can use chip that explore like hundreds of thousands markers all over the genome, and you can see if one loci is, or if one locus is methylated partially or totally, and you can also compare every genes in the genome. So that's something we did during my PhD. We look at the longitudinal change of DNA methylation during the emergence of psychosis. So we took advantage of a cohort of at-risk mental state patients recruited in St. Anne Hospital, following during 1 year. Some of them remain at risk at the end of the follow-up. Some of them develop first episode of psychosis. We call them converters. And we compare the DNA methylation in both groups longitudinally. It means that we did a difference across time, and we compare both groups. And we found a list of 5 genes that are differentially methylated across time. And for instance, we found genes related to like CHL1 or NRP1 axon guidance, or GSTMM5, for instance, is a gene involved in redox mechanism. But DNA methylation is related to gene expression. So DNA methylation is correlated to the expression in terms of messenger RNA. So the next step was to explore the messenger RNA using the same design. We did that in exploratory cohorts of 15 at-risk patients using RNA sequencing. Then we, in the same cohort, we did a confirmatory study with 31 converters, 63 non-converters. And we found some interesting genes differentially expressed during the onset of psychosis. The first gene, CPT1A, is related to fatty acid oxidation. This is of interest because these genes can be modulated by omega-3 fatty acid, and there is a increasing literature on the role of fatty acid in schizophrenia, but also as a potential targets to prevent psychosis in ultra-high-risk patients. Unfortunately, in the last NeuroPro study, which is a big study led by Patrick McGorry in Australia, the use of omega-3 did not totally prevent the onset of psychosis in ultra-high-risk. But that's still an interesting story, because maybe in this study, the observance was not perfect, and in the same time, maybe they just prescribe this kind of pills to a large amount of patients, not taking into account the difference in terms of omega-3, omega-6 balance in different patients. So maybe if we restrict that to some subgroups, the benefit could be better. The second gene of interest will be the GSTM5, which is involved in oxidative stress, because there is antioxidative drugs. And the last one, neuropilin-1, who is involved in neural migration, axon guidance, and I don't have many targets for these genes, many clues to use it as a targets, like a treatment. Okay, so what we are doing now, we are trying to complement these first studies of gene methylation and gene expression with exploration of other epigenetic mechanisms, and we focus on macroRNA. So macroRNA is small RNAs that could modify the expression of genes, and we, using the same design in a larger cohort, we also do near-seq analysis, which is basically we sequence all macroRNA in the blood, and we compare the individuals depending on their outcome. So in this analysis, which is unpublished yet, we compare individuals' macroRNA expression and change across time using a differential macroRNA expression. And we found that some interesting macroRNA are dysregulating during the onset of psychosis. You can see here a zoom on the MIR155P, which is a macroRNA which is similar at the beginning in both groups, but decreasing during the onset of psychosis, only in the converter groups. The non-converters who did not develop schizophrenia have a similar level of expression before and after the follow-up. The second analysis we have conduct on this macroRNA is a supervised machine learning story. We are able to predict the onset of psychosis with a area under the curve that is 66%. That's not too bad. That's not perfect, of course. But the most interesting story is that we show that there is no overfitting, and we can identify the macroRNA contributing to this prediction algorithm. So we keep a list of this candidate macroRNA for a further analysis. The last story is a network analysis, because it's not only a question of which gene can predict, which gene is differentially expressed, or macroRNA is differentially expressed. The story is also how did the macroRNA work together inside of a network. And when we are working on a network analysis, there is two different conception. One is the hub, which is quite easy to understand. Hub is a gene or macroRNA where all genes are converging. So it can be measured by the degree centrality. All the genes here are related to others through the hub. Another concept is the bottleneck. The bottleneck measured by bit-witness centrality is a way to detect the amount of affluence a node has over the flow of information in the graph. So it means that this network here with the hub G is related to other genes through the bottleneck. So the bottleneck also carry some information. If you decrease the expression of the bottleneck, you will totally change the relation between two parts of the network. So we construct a correlation network in converters and in non-converters. And we found that the patients with conversion to psychosis have more, finally, have a higher bit witness and lower degree centrality compared to the other group. It means that the macroRNA used during conversion to psychosis are more related to bottleneck than to hub. So we identify also a list of macroRNA, and the last step was to compare the list from all three strategies, and we end up with a list of 25 candidate macroRNA listed here. So the macroRNA are controlling or changing the expression of many genes in the genome. And we can infer that based on some algorithms. So we end up with a list of microRNA. We enter this list to software able to predict the gene targets of this microRNA. And we submit this gene list to enrichment analysis. And we found that the genes dysregulated by microRNA during the conversion to psychosis are related to general cognitive abilities, intelligence, but maybe also to schizophrenia and other kind of psychiatric disorders. So this is a way to show that this is not by chance that the microRNA are dysregulated. These microRNA dysregulated during onset of psychosis are related to genes involved in some important pathway. And this pathway, we can also look using some gene ontology term, and we found that the genes are more than by chance related to oxidative stress, membrane trafficking, and also neuron guidance. So again, we are converging with this third analysis to oxidative stress. So the last point was to look at the oxidative stress markers directly during the psychotic transition. What we did is we compare converters and non-converters using a logistic regression, and we put many markers of oxidative stress in the equation, directly measured from blood, like the glutathione peroxidase, for instance, the glutathione reductase, the T-bars, PRX4, which is the peroxyredoxin 4. And we found that this PRX4 marker is different between converters and non-converters during the onset of psychosis. So the change of this expression of this enzyme is different, and that may confirm that oxidative stress is important during the psychotic transition. Oxidative stress is related to aging, of course. So we come back to the epigenetic data and the DNA methylation data, because DNA methylation can be used to do epigenetic clock, so to measure the aging of the patient. And as we have a longitudinal design, we compare the different aging across time between both groups. And what we found is that the converters age more than non-converters during the onset of psychosis. So during 1 year of follow-up, non-converters they age from 1 year, non-converters age for 2.5 years. So they are aging more because there is more oxidative stress in this group. So why did they age more? So based on the previous story is because they have more oxidative stress, there is dysregulation on that. So what are the consequences in terms of brain aging? So we take advantage of the MRI data, imaging data, and we compute two factors. One is total surface area, which is not changing so much across time. The other one is the mean thickness variation. So the thickness of the cortical thickness is really the measure of the thickness of the cortex. And we plot the cortical thickness for each individual based on their age. And we found that the ultra-high-risk converters have a slope which is decreasing across time more than the non-converters. It means that it's not a real purely longitudinal study. It's like a pseudo-longitudinal where we plot different transversal measures based on the age and suggesting that the redox mechanisms are imbalanced during the conversion to psychosis and have, as a consequence, an effect on cortical thickness. So maybe the onset of psychosis is related to redox mechanism and accelerated aging. This is of interest because there is some study exploring the role of N-acetylcysteine, for instance, to prevent the onset of psychosis. And in this story, a patient with first episode of psychosis had been treated by N-acetylcysteine, which is an antioxidant agent. And what the colleagues Philip Conus and all show that there is no clear effect on the PANSS, which is a scale for psychotic symptoms, nor for the functioning symptoms, but there is an effect on the cognitive function of individual with first episode of psychosis. There's an effect of the verbal fluency test, making test, and processing speed factors. And it might be also an effect on the connectivity in the brain, especially the fornix, which may be beneficial based on N-acetylcysteine supplementation. So now with Mario Krebs, which is the head of my team, we are involved in a large clinical trial, where 500 patients should be included in the next years all over France, and the patient will be randomized into 4 arms. Patient will be ultra-high-risk patient, first episode patient. We will treat them as usual for some of them. In the second arm, we will treat them as usual, plus we will add a cognitive remediation. In the third arm, we'll add personalized treatment. So for instance, patients with deficit in omega-3, we will give them omega-3. Patients with high redox level, we will give them redox supplementation. And in the last arm, who will be maybe the most beneficial for the patient, we will treat them as usual, plus cognitive remediation, plus specialized and personalized treatment. So this is a big challenge for us to be able to intervene early, but also to give treatment that could be acceptable for the patient, and sometimes that could help for reducing the symptoms or even maybe prevent the onset of psychosis. I would like just to finish with some acknowledgement to the team. Of course, it's work done by many colleagues in Paris and all over France too. Thanks a lot.
Well, thank you very much, Boris.